through it, was looking at the titration curve for tyrosine. Remember, we did the titration curve for just a general acid, well, however many days ago that was. I think last week, Monday, something like that. And so I noticed that some people seem to be a little freaked out by that, which is perfectly understandable because of different backgrounds. Okay? So that's one of the reasons why we went through it with the general information first, and then we see it again with our amino acids. Okay? So it is something you're responsible for in a biochemistry class, and it is unfortunate that some higher power said that you didn't need to have that chemistry background to do it. Ultimately, because what they ended up going through and doing was something that I similarly say, you have the content in biochem, almost all of it is memorization. So what I will go through is the derivations on where things come from. Because even though I have a biochemistry degree, my memorization skills are awful. And so I always have to derive things. I have to break it down to first principles. Right? Which means as a biochemist, I'm pretty horrible because biochemists need to have things memorized and kind of at their fingertips. Okay? So the general strategy that I had for almost every exam was cram as much information in as possible, and then as soon as you get the exam, purge as much information as possible and hope for the best. Okay? And that's, I mean, that's a biology test. Okay? It's that same kind of principle. Okay? So if you had, I mean, most of you have probably had anatomy and physiology. That's kind of the impression that I've been getting those classes are too. Okay? And yes, we have some applications within that, but it's a lot of just facts. Okay? You aren't really applying those. So be aware that while we are going through the derivations in class, you're not responsible. I'm not going to sit there and watch you and I'll say, derive this for me. Explain why each and every single step. What you're going to be responsible for is drawing that. Okay? And I might ask a multiple choice question. What is the pKa or the first pKa for this random structure based on this titration curve? You should be able to do some of that interpretation, but if you have this memorized, or if, say, somewhere earlier in the test, I asked you to label a titration curve, you now have the answer there. Okay? You just have to go back and say, okay, I put all this information on here, how do I interpret it real quickly to answer this multiple choice question later in the exam? Kind of make sense? So use the exam to help you out. Okay? You will see multiple choice questions that ask kind of derivations off this. Okay? You don't have to derive them, you'd have to have it memorized. So you either draw it all out, or someone already set you up to do that already. Okay? Kind of make sense? Okay. So all of the information that I would expect, or that I could reasonably expect, is in this right now. Okay? I did make some abbreviations, just like we did with the acid. We did HA, or H2A, or H3A. Okay? I did simplifications of that with this structure. We've got our reaction running along the top with tyrosine, and I went through and specified the charge state of each of those tyrosines. If you're following off the lecture slides, remember that very first structure had a massive typo because it said O- for the carboxylic acid, and it must say OH. Okay, so it is now fixed on my slide, but it's not, or fixed on the image up here. It's not fixed on the slides. Okay? And if we evaluate the charge state, which is going to become important for, I think, the very next slide, okay, we can see that the charge state will change. Under acidic conditions, we should be very, very positive because, well, acids are H+. So if we're under acidic conditions, we would fully expect a positive charge. And if we're under basic conditions, we would expect a negative charge. Okay, and that just comes back to kind of applying the definitions of acids and bases. Okay? And we could, again, we don't have to have that memorized if we've recognized the pattern within our structures. Okay? Kind of makes sense? Okay. So that's what's happening as we slowly increase the pH. When we look at our titration curve, typically we start with the most acidic form and we slowly titrate in base. And initially, again, what happens is we add the base. Okay. The pH changes slightly. Okay. It almost always goes up kind of at a pretty steep curve initially, and then it kind of levels off. It does not level off as much as I've got it here. Okay. That's because I'm hand drawing this, but it levels pretty consistently. Your pH change is usually really, really minimal. Okay? And as we add more base, what happens? Well, the acid form of our amino acid, the positive one, starts losing that hydrogen. Well, if it loses a hydrogen, it then starts to become less positive. Well, what is less positive than a +1? Neutral. So eventually we make it to the stage where it's all entirely neutral. Okay? And I did hear a question about this one because it pops up in the homework. Okay? If this is the curve that we've got, when is our structure +1/2? Yes. Okay? And the typical thing is you look at the charges and you say, okay, well, it's never +1/2. Well, then that's a really hard question. Okay? Because they're saying at what point, at what pH. You have to enter that value. I think you have to enter that value. So it does happen. The question is when and how are we determining that charge? Well, we're not saying a charge on a single molecule. We're saying a charge on the overall solution. Okay, so what do I mean? What happens to the charge on the overall solution if we're purely acidic? What is the overall charge of our, it's +1. What happens if I add one molecule of base? What happens to the positive charge? Becomes less positive. I've removed one of those positives. So it starts to transition towards that 0. It starts to transition towards +1/2. When am I going to be at exactly half? The half equivalence point between those two. Okay? And it doesn't have to be a half. I could have said +3/2. Yes. +1 or numbers. -1/2. What would the pH be to be at -1/2? Okay. I'm looking at the point when I transition from a -1 to a -2. So -1/2 is between those two, which ends up being the third half equivalence. Okay? So we can pull kind of odd information off of it. Why is the homework necessarily asking you to pull that information off? Because it's asking you to interpret what the overall thing is saying, not just saying these individual points. Okay, so they're trying to get you to think a little bit more about it. So it's a good question in that respect, but it makes it much more difficult to answer. Kind of make sense? Okay. Big things to address. Your half equivalences are hugely important because your pKa equals your pH. Right? That's massive. Okay? So that's really, really, really important. You need to know that information. What else is important to pull off of this? Which is actually not labeled as a hint. Should I label it as a hint? The buffer range. Okay? We do have ranges for the buffer as well. Okay? Which is going to be roughly where it stays as flat as possible. Okay? We could conversely say, where is the worst place to be a buffer? The whole point of a buffer is that what? The pH doesn't change. So the worst place is anywhere it's changing. So you could reference any of the equivalence points as being very bad places to run a buffer system, because it won't buffer. Okay, kind of makes sense? I actually kind of like that question. You might see that one. Okay? So we good interpreting that for the most part? This is now the third time we've kind of seen it. Granted, the second time was stupidly fast. The first time was reasonably paced. Another question that we could pull off of it that I think comes from the online homework. What happens if, say, you were given this curve, but you were not given that? So what did I erase? Molecule. So what could I ask now? and not the generic formula, because we could do the H3. That's not a bad idea. Okay, this pushes more towards the homework and less towards an exam. What's that? What do you mean, push that a little bit further? Because that's what Ray was kind of saying. How do you know what the structure would look like? So we're getting at the right idea. Yes, we're looking for some kind of structural information, and it's good that you brought in the pKa. What structure should I draw? What if I drew... I need to get more glue. Pen's breaking. I think I can draw it here. Whoops. Anyone want to help me out? What was that? Aspartic acid? I think? Or it's glutamic acid. It's one of those two. I think it's aspartic. It's glutamic. So it may be called glutamate or aspartate. No? Man, you're still saying I'm wrong? That's no fair. I don't know. I could easily be wrong. No, I was right. Cool.
Okay.
Aspartate. So
What did I?
glutamic acid, aspartic acid versus aspartate. Sorry, aspartate versus aspartic acid. Why the different name? It's protonated versus deprotonated. Why did they change it on you? Just to be irritating. That's all I got. Okay, so this form would be the aspartic acid form. And if we got rid of structure underneath, got rid of that hydrogen, it's now aspartate. Okay? Because it's not officially an acid anymore, so we can't call it aspartic acid. Okay? So the name will change depending on that protonation state. Okay? So would that be a valid structure for this titration curve? Okay, so there we have neutral, so I'm saying that that structure exists at this first equivalence point. Right, the neutral one. Right, it's neutral, isn't it? Isn't the first equivalence the 0 point? Is that correct? And no. You know why?
I think the H on the second carboxylic acid group would come off at a higher or lower pH.
So looking at the titration curve isn't just labeling H3a, okay, or drawing a structure. That is a structure. That structure does not match this curve because, as you were suggesting, the pKa's don't match. So when we look at a titration curve, because we've memorized all of our amino acids and all of their pKa's, we would say this can't possibly be that one because the pKa of the carboxylic acid, this one we can kind of cheat on. The pKa of our acid functional group was 2. What's the other acid functional group? Probably pretty close to 2 because its structural difference is not that far off. Okay? So we have 2 pKa's about 2 and 1 about 9. Is that what this curve shows? pKa at 2 and a pKa at 9 and 10. This curve does not match that structure. Okay? So the question that could be asked in the homework, which it is what is the amino acid for this titration curve? You have to interpret the pKa's off of it and be able to get to the structure. And that's where you might say, oh my God, I have to know all the pKa's. No, that's why it's a homework question. You're expected to look up those pKa's and match it because it's coming from that interpretation. I don't expect you to memorize it for the test. Could I still sneakily, backhandedly ask it? Yes. I could give you this curve, and since I know at least one of you has an amino acid chart, okay, handy, I could give you this curve and say for which of the following, oh, I can't do that because I don't want to draw the structures. I would draw the structures, number 1, okay, and I can't draw all the structures. Glycine, alanine, tyrosine, isoleucine. So 4 answers, because I don't want to come up with a 5th. All right, should we write those out? Answer choice A, glycine, that says glycine, Answer choice B, alanine, C, tyrosine, and D, isoleucine. Okay, so you all know the answer, right? Okay, it's tyrosine. But how would you process your way through that? Remember, given the structures. I don't expect you to look at the names.
The different R groups.
Look at the R groups. Okay, and Mark? Yeah? Mark? Yeah, okay. You just didn't look up again. Made me nervous. Okay. When you look at alanine, how many acid-base groups are there? There should only be 2. Well, I have 3 half equivalence points. Can it be glycine? No. How about alanine? Again, only 2. Isoleucine? Only 2. This curve can't match those 3. So I can still ask it, but I'll ask it in a way that didn't require a memorization. It required an interpretation of the structure, looking for those acid-base functional groups, because we've talked about those. Does that make sense? Okay. Realize in the process of asking that, that means I have to draw all those structures, and that's a very tedious, time-consuming thing. And so typically I don't ask questions like that, but again, I kind of like that question, so you might see that one. Yes? Yeah.
So how do you know where the equivalence is?
So the equivalence is where your pH spikes, because what's happening, so that comes down to the definition of what the equivalence is. Okay, your equivalence point is the point where the moles of your starting acid equal the moles of your titrating base.
But it's not like a number, or just a something.
It would be experimentally determined. You wouldn't just look at it and say, look at a blank thing and say the equivalence point is here. Okay, so you'd be looking at a titration curve. Your equivalence point is going to be, it's mathematically the inflection point. If we look at this part of the curve, it's concave up. If we look at this part of the curve, it's concave down. How did we transition between those? We went through the inflection point, which is our equivalence point. Okay, mathematically. As far as chemically, the equivalence is just the moles of this equal the moles of your base. Okay? And yes, that could theoretically be calculated. That involves numbers, exponents, scary stuff that I don't want to deal with. Okay, so does that sort of answer your question? Okay. Other questions on it? Okay, so let's bring, and I erased all that structural stuff up at the top. Okay, if you maybe remember when we ended class when really the clock ended class, because I kept talking, and really when the next class started walking in, because I kept talking. And we looked at our structures up at the top. We looked at the structures up at the top. And I had all those charges in there. We had what? We had +1, we had 0, we had -1, and we had -2. And I said there was an important point at which we wanted to evaluate and look at or represent our structures. Okay? And that was the neutral point, okay, where our structure was neutral. Okay? That is such kind of an important chemical value that it is something that is very often reported for our amino acids. Well, if it's reported, we have to know how it was reported. That's our calculation. Okay. How do I know when I have just the neutral structure? There's no charge. I get rid of, I can't quite get rid of all of those. I want to know the point, the exact pH at which I have the neutral structure. So looking at the structure doesn't give me any information about the pH. What else would I need? So we need a couple things. We could, or a couple routes. We could do an average of the pKa's around it. So by looking at this, we could take the pKa's and average those two values. Why can we get away with averaging those? Well, let's go back to the titration curve. When do I have just the 0 structure? Okay. I know there's a lot of stuff on here. I'm going to try and pick a new color-ish, purple. This is when I have just the 0 structure. I want to know at that equivalence point, what's the value? Okay. Well, I could guess off of this curve by interpreting the y-axis. Okay, but we can do better than guess. Because we are titrating in the exact same amount of base each time, that equivalence point is exactly halfway between the first and the second half equivalence point. Okay? Well, how do I know what the value of the first and the second half equivalence points are? Because that again means interpreting it off the y-axis. The pKa equals the pH. If I'm told what the pKa is, I then know the pH, the exact value of the half equivalence, which then means I can average the value quickly. That's why that formula works. So we take the average of the pKa's around the neutral structure. Which means in the case of tyrosine, I take the average of pKa1 and pKa2. What do I do with pKa3? Nothing. Okay. So by looking at that structural setup, I can evaluate, first off, our derivation is recognizing why we can use those pKa values. It's halfway between our two half equivalents. Okay. Now all I got to do is look at the structure and assign have my pKa values for those, and just average the pKa's around the 0 point. And that's it. That's it. Quote unquote. Okay? That means we have to do a structural interpretation. Okay? How do you do the structural interpretation? Well, number one, what do you need to start with?
Structure.
The structure. Okay? This is one of the reasons why most biochemistry classes insist that you memorize all of the structures, because you need to be able to do that interpretation. Okay? So for the sake of the exam, what I would do is probably give you a structure and say, give me, and here's a bunch of pKa values that potentially match this. Okay? I won't tell you necessarily where they match up, but if I gave you 2, 9, and 7, where's the 2? The carboxylic acid, the 9, the amine, the 7, the R group. Okay? That's why you need to have those kind of pieces associated appropriately. Okay? So with that, you can then start to interpret something about the charges. Okay? This gets us what's known as the pi point, or the isoelectric point. It's the point at which our structure has no net charge. Okay? That does not mean it's not charged, it just means it's no net charge. Okay? Most commonly, for all of our amino acids, the pi happens to be what value?
6.
For a little bit lower, 5, right? Okay, it's halfway between 2 and 9, right? Because it's the average. Okay? I'm not the greatest with averages. I'd probably pick 6 and I'd say 2 is 4 units away from 6, and 6 is 3 units away from that. It's a little bit lower than that. It's probably 5 something. Okay? Or, how else can you do calculations? Calculator. Okay, you're more than welcome to use a calculator. So for most of our amino acids, the pi is obvious because it's just the average of the two pKa values. And if okay, what happens when we move to the bottom 6 or 7?
The charged R group.
We have the charged R group. That means we have a third pKa. That means we really have to evaluate the structure, decide when it is neutral. Okay, I can't just pick any 2 arbitrary ones. I have to evaluate the structure. Okay? So that's why we had to have drawn out our tyrosine, looking at our structures. Again, we have that typo. It's really starting to get irritating. Clearly not irritating enough for me to fix. I would look at the charge states for each of those, find the neutral structure, average the 2 pKa's around it. Make sense? Okay. If I switch to a different amino acid, well, I switch to a different structure and I do the exact same process. Okay. The textbook, I just kind of copied and pasted this out of the textbook. It confused the hell out of me. I have no idea what they're trying to say. I think I finally nailed it down because they give you this formula. Say pKa1 plus pKa2 over 2. Does the 1 mean the first pKa? No, it just means arbitrarily one of the pKa's. Yeah, like really? Okay, but yes, that's what they're saying. It's not pKa1, it is the first pKa you decide to average. pKa2 is not the second pKa, it's the second pKa you decide to average. Okay? So that when you then go through, it's saying pKa1 for the function group that is associated at the isoelectric point. So that's a fancy way of saying the pH is higher than the pKa. That's why we removed the hydrogen. Okay? So all of that text is really telling you what I just told you to do with the structure. Okay? I have seen and heard of another method that has to do with, well, if it's a basic amino acid, you use these pKa's. If it's an acidic amino acid, you use these pKa's. Okay? So really they're asking you to memorize a bunch more useless stuff. Okay? Stick with the structure interpretation. The derivation really isn't bad. Okay? If you were a full-time biochemist, then you would probably just have the value memorized. You wouldn't even bother to calculate it. You just know what the PI was. Okay? Which brings us to a nice big summary of everything. Isn't this fun? We've got our name, 3-letter code, 1-letter code, pKa for the acid functional group, pKa for the amine functional group, pKa for the R group, and the PI. All in one glorious, fancy chart. Thank you, Internet. Okay. Officially, what you should be doing is picking 5 and getting the name, 3-letter code, 1-letter code, and the structure memorized, along with pKa for the acid functional group, 2, pKa for the amino functional group, 9, and then if you want to memorize the third for 1 or 2, that's fine, you can do that, but I'm not expecting you to memorize the third pKa. Kind of makes sense? So 5 amino acids, structure, name, 3-letter, 1-letter, and at least the pKa's for the acid and the base. Or the acid and the amine, I should say. Okay? We okay with that? At least as okay as we can be? Okay. Remember, doing the PI and the structural analysis or analyses still comes back to that balance of the pH versus the pKa. That is a very important analysis that helps us kind of adjust what happens to our charge state of our structure. Okay? Proton on, proton off. Notice we're using HA and A minus. That's a bit misleading because the amine starts as its protonated state as HA plus, okay, because it's basic. So that's a bit off. It's just a shorthand. Okay, protonated, deprotonated. So some of the big basics to remember out of our amino acid chapter. We have 20 alpha amino acids. What does alpha mean? What's that? The first, yes. What do you mean by that?
It's with the amine groups attached to the alpha carbon.
If we look at a standard structure, okay, back to OChem, if we had a carbonyl here, what is this position referred to as? The alpha carbon, because it is the first carbon from our carbonyl. So it's the alpha. The next one would be beta. The next one would be gamma. It's that symbol. Okay. Do we have amino acids that form from beta and gamma? Yes. Did we look at any of those? Why not? It's not resonance. Right. Here's our general structure. Bless you. First position is alpha. Where's the next position? There isn't really one. Our beta officially goes into the R group. Okay. We don't tend to see amino acids like that. They're very uncommon. Do they exist? Yes. We don't stress about them because we don't encounter them often enough to worry about it. Just realize that they can be there. So there are 20 alpha amino acids that are super, super common. 19 of those 20 are primary aminos. Okay? Meaning, when we look at the amine structure,
It's single bonds.
not quite. What does primary mean? It's only connected to one carbon. There's one carbon bond. That's a primary amine. It says 19 of 20. Proline is not. What is proline? What happened with our proline structure? The R group comes back and bonds to that nitrogen, which then means I have a second carbon attached. It becomes a secondary. Okay. 19 of the 20, the alpha position is a stereocenter. Okay. Usually L, though it can vary again depending on the conditions, but very typically it is an L amino acid. Glycine is the only exception. Why is glycine an exception?
H is the R group.
The R group is an H. Okay. If we look at the amino acid that's drawn up there, for it to have 4 distinguishable groups at our carbon, we need that R, nitrogen, and this C to all be different from whatever's back here. Well, what's back there? A hydrogen. They all must be different. If the R group is an H, are they all different? No. So glycine, which I always get confused with alanine. Anybody have a good trick on that? Remember glycine is the one with the H?
Phenylalanine versus alanine is just phenyl group.
That might work. I'll try that. Okay. Glycine's our exception because we don't have 4 distinguishable things. So no chiral atom. Isoleucine and threonine are even more fun because there's a second stereocenter in the R group. Okay. We didn't really talk much about that, but we just have to realize that they're special because of that extra R group. Okay. Uncommon amino acids are all derived from the common ones. So we could switch between L and D by doing some chemistry, removing some hydrogen, shuffling some stuff around, putting hydrogen back on, and switching that stereocenter, depending on the direction. Why would we do that? Okay. Do I want a nail only having one function, or could it have multiple? Multiple, depending on what I'm looking at. Very typically, I use a nail to put 2 pieces of wood together. Okay, but I could use a nail to, I don't have any hair, hold up hair, a little pin thing, right? Not putting it into your head, but like a little spinny thing. Okay? So there are other purposes for it. It just depends on what we need. The uncommon ones all come from a common one. Okay? And then the last big thing, your carboxyl, that's your 2, and the alpha amine is 9. Okay? Again, numbers, I don't remember the 2.19, I just remember 2. And the alpha amine is officially 9.5. I only remember 9. Depending on what level you're working towards, you may need more resolution. Okay? A couple of you are looking at MCAT stuff. They might want more resolution. I don't know that. Okay, but it could be 2.2 and 9.5. Kind of makes sense? Okay. Yeah, almost. Almost out of chapter 3. So our peptide bond, the peptide bond is linking to amino acids. Okay, so we've seen this slide before, so let's go through and identify some extra pieces out of it. We've got the pretty stuff that your textbook goes through and draws, but then removes useful information that we would get out of just organic chemistry. What type of chemical reaction are we doing? Lots of names can come out of this, by the way.
Dehydration.
We could refer to it as a dehydration, because what changed to the structures we move across? There's a loss of water, so we could call it a dehydration. The issue is dehydration very typically refers to the formation of alkenes, and so it's not quite the greatest, but it's a possibility. What else? Just include it. Dehydrate. What happens to that water? Okay, to do this reaction, we're generating lots of heat, so the water turns into a gas, right? Okay, so it evaporates, okay, and rises up in the system. Rises up away from where the heat is actually located, which then means what happens to water when it's, or sorry, water gas when it's no longer heated? It condenses and we have a condensation. Right? This one's really only going to go to Jen. Sorry, Jen. Could be put on the spot. Mechanistic approach. Maybe everybody else can figure this one out too if you went through OChem. What is the mechanism?
Isn't it like the substitution?
No. It's an addition elimination. Okay. We end up adding the nitrogen to the carbonyl. So there's the addition, followed by the reformation of the carbonyl and the kick out of the OH. That's not how you spell elimination, just in case you were curious. And that's not getting any better. So let's just do that. Addition and an elimination mechanism. Okay. What type of bond is formed? And actually, let's take a look at the top, our product over here. Where's the bond formed? It is called an amide. Carbonyl where?
Carbonyl carbon.
So,
And the amine, or the N.
and the amine. So what we're looking at right there is the amide bond, which is often referred to as the peptide bond in biochemistry. Notice that we have some other pieces out there. What else happened? We have another amide bond. What does that mean? Polymer. It continued to react. What does the squiggly line mean? This structure continues outwards. Okay? So that's representing our polymer. So in this case, I didn't do just a single reaction. I said this is the basic pieces, and then that could continue onwards. Which amino acid did I draw? Wasn't alanine, right? Was alanine? Oh, son of a... I don't remember. I was getting backwards. Which amino acid I do? It's the one with just hydrogen as our group. Glycine. Frick nuts. Can't get that one right. Okay. The type of bond, we've got the amide, the peptide. Any interesting features in that? Okay, so let's clean up that bond. Let's look at it up top because I think it's easier to see up top. Draw brackets around it. So we're talking about that line. Do you notice anything interesting there?
You can do resonance.
We have implied lone pairs on the nitrogen, which can do resonance, which suggests what about the strength of that bond? It's pretty strong. Okay. We're looking at oscillating between a single and double bond. Okay. What else does that mean about it if we're oscillating between a single and a double bond? What's a characteristic of double bonds? Pi bonds. What's characteristic of pi bonds? Yes.
Sterics.
Sterics does kind of play a role in this. The sterics we'd be looking at, because again we're focused on that peptide bond, is we're looking at that group versus this group. So in this conformation we'd be locked in the, let's avoid E and Z, that's just nomenclature. The simpler version of it.
Cis and trans.
Cis and trans. We're in a trans conformation because our large groups are aimed away from each other. Okay. So if we have a double bond or a pi bond, we have this cis-trans issue, which means what? Why do we refer to cis and trans? There's limited rotation about that bond. Okay. We can rotate because it's not formally a double bond, okay? But there's limited rotation there. Why? We want the resonance with the nitrogen in. If I start to rotate, I lose the resonance ability. The p orbitals no longer overlap, and I don't have that double bond. I lose the ability to do resonance. Okay? So a rotation about that bond can change a lot of chemistry about that functionality. Okay? And again, note our standard notation. Left is the amine, right is the acid. So if we look up polypeptide chain, it's always going to start nitrogen on the left, acid on the far right. Okay? Does Japanese read right to left? Always wondered that, like if in a different language, do they then interpret the opposite direction? Probably not a good question to ask now. I apologize. Left to right, left to right. Amine acid, amine acid. Okay? So let's put this all together in a very happy little slide. Given a peptide chain and a pH, let's say pH 3. What can be determined? So what could be a possible question that I could ask? That is a valid amino acid sequence, by the way, so don't give me anything for that. I'm just being a scientist. What is S? Serine. What is C? Cysteine. A? Alanine. R? Arginine. I like that one. Arg. That one. That's the only one I can remember. Y? I thought that was tyrosine.
I'm making guesses.
Oh, you're just guessing? Okay.
Yeah.
S? Serine. H? Histidine. I? Isoleucine. T? That's threonine, I think. That's threonine? I'm not positive on that one. Okay. What could we determine? You ready for the awesome question? This is everybody's least favorite question. Talked to somebody in one of the biochem classes, and this was already in my slide, so it's not like I talked to them and was like, let's do this. Okay. Talked to somebody in one of the biochem classes at ASU. They get this question in 3 formats. What is the charge on the peptide? Single letter code. 3-letter code, structures. And you have to determine the charge on every single one of them. What do you have to do to determine the charge on that peptide? You have to look at the structure. So outsides. Why are you saying just the outsides? What's on the outside over here? The amine, right? Okay, we have the amine out here, which could be protonated or deprotonated. Okay, so that is going to contribute to charge depending on the pH. So that's a pH variable. Okay, what's out the other end? The carboxylic acid, which is also pH variable. So according to Ray, this isn't that bad. All we got to do is look at the ends. That's easy. Also have to evaluate the R groups running through the middle. Okay? This is where having some of the having the amino acids memorized in general makes this a little bit easier, because then you can start to interpret the structural stuff. If you're given just the 1-letter codes, you have to be able to interpret the structure down. Okay? So if we looked at serine, cysteine, alanine, arginine, so tyrosine, that was our serine. Histidine is going to be our tricky one. Histidine is a variable one that has an R group with a variable pKa. So does tyrosine. We said that was tyrosine. Okay. So we would then have to tag those as R groups with variable pHs. Okay. The histidine's the big one. Tyrosine's a little bit less so. The isoleucine and the threonine, we don't have to worry about, because those are just our nonpolar aliphatic chains. Okay? So we would then look at it and say, okay, if I'm at a pH of 3, what happens to the R group? Okay, well, we just looked at the R group for tyrosine, so you all memorized that one already, right? Is that a no? I guess that was a no. Our pKa 3 was 10 point something. Okay? So at a pH of 3, what is the relationship? Am I more acidic than the pKa?
It's deep, or it's protonated.
It's in its protonated state. Okay, but again, we have to be careful. Are we looking at an amine, protonated state being positive, or are we looking at an acid, protonated state being neutral? Protonated is neutral. That's why I put the slash there, or try to specify that. Okay. Anybody remember our tyrosine structure? It's the phenol. The protonated state is OH, which means neutral. So it is not contributing to our overall charge. What about the histidine? I don't have that memorized at all. Make things up. C6, damn. Just trying to throw out some letters and seeing if it worked. Okay, so we're about 6 for our side chain. Are we more acidic or less acidic than our pKa? More acidic. So are we protonated or deprotonated? Protonated. What is the side group? It's an NH that's matching over here, which means the charge on our histidine at pH 3 is positive. So that means our overall charge on this molecule should be positive, right? Did we evaluate the amine and the acid yet? No. What happens to the amine? Okay. We're at a pH of 3. The pKa of the amine is 9, so it should be protonated. The charge contribution from that is positive. How about for the acid? That's probably going to be negative. That's a bit close to actually test on, okay? But that would likely be negative, which means our overall charge for the molecule is, it was positive. Okay? Fully expected to do this in the homework because you have access to all those values. If it shows up on the test, it'll be a much shorter amino acid sequence, and you'll be given a lot of data to match it up because I don't expect you to have that memorized. Yes?
So why didn't you do like the C, the A, the R?
Okay, so let's take a look at it. Ooh, this could be embarrassing. I think I can get away with it. The cysteine. Did I do cysteine right? Yes. What is the pKa for the R group? So we go through and look at our tables. What is the pKa for the R group? What's that?
8.33.
Son of a... Oh, well, it's not going to be that big of a deal. The pKa for that R group was 8.33 or 8.3. Protonated or deprotonated? Well, you're still at a more acidic state. The protonated state is SH, so it didn't contribute, so that would be a neutral one. I was hoping that I picked a better one, but I don't remember all the structures. Alanine, that's the CH3?
It's a methyl.
Okay, this is more along the lines that I wanted. What's the pKa for that R group? I can tell Jen knows this answer. You better know this answer. Okay.
Don't remember.
There is no R group according to the table. Yes, it does actually have, or sorry, not an R group. There is no pKa. It does have a pKa. It's like 50, meaning it's not going to contribute. Okay? So we don't look at the other ones because according to their pKa values, they don't contribute. They're not going to be anywhere near a standard pH range to evaluate. We can look at them. The pKa for alanine is 50. Okay? Its protonated state is there, CH3. What if I'm at pH 10? Still more acidic. Okay? pH 30. Okay? Well the hell off our scale. Stupidly basic. It's still more acidic than the pKa here. Okay? So some of our R groups, their pKa's are so high that they're never going to hit the pH range to worry about, so they will never change their structure. That's all the neutral nonpolar, that's the word I was looking for. Amino acids, we don't worry about. Okay, so the only ones you have to worry about are ones that are, and sorry for kind of blurring, for that kind of assignment, whoop, come back here. Oh, I don't like that one. The only R groups you have to worry about are the ones that have a third pKa value. Okay? Because the amine and the acid of each of those in the middle, well, they're gone. They're locked up in making the peptide bond. So I don't have to worry about them. The only ones I have to worry about through the middle would be the R groups. So if they have a third pKa, then I need to worry about it. If they don't, I don't worry about it. Does that get your question? You don't quite look satisfied with that, but okay. Let's bring up the question about the amines and the acids in between. We said we had to worry about the amine out here. I'm just going to draw it as NH, and the acid out here. I'm just going to write acid. Why do I not worry about the amine on this side of alanine and the acid on this side? They're locked up in the peptide bond. When we look at the peptide bond, there's our acid. Where is that? Here. There's no more acid. It was eliminated from the structure to make the peptide bond. Okay. I do have a nitrogen here. So couldn't that nitrogen act as a base? What did we say was special about peptide bond? Resonance. To do resonance, what do we need? Electrons. Are those lone pairs available to react as a base? Nope, they're tied up in the bond. So any amino acid running through the middle of our peptide chain has no effect, or the, whoa, where did you go? There it is. Has no effect on our pH, or on our charge state. The only thing that will have an effect is what's exposed. That's the R group. So we're really just looking at the R groups for each of these. I did a big skip because I said, well, I know that those don't have a third pKa. So I'm just going to ignore those. The only ones I have to worry about are the ones with a third pKa. Does that get a little bit better, maybe? That looks a little bit more of a confident head shake, yes.
So you have to memorize those R groups then?
You would need to know those R groups to answer this question. So, and I didn't say memorize there. Okay, so here's the reason why. On the homework, do you need to have it memorized? No, because you have it. You're expected to look at those. On the exam, would you have to have it memorized? No, because I will give you that information. Okay? I won't just leave you hanging on it and say you had to have that all memorized. Okay? Because upper, lower division levels. And if it comes down to it on an exam, you're like, you said at this point in this video that this question wasn't going to be on here. I'm like, yeah, I think you're right. Here's the points. Take it back. Okay? So always feel free to challenge it. And yeah, you're right. For this kind of thing, you would have to have it memorized, but you're not at that level, so don't worry about it. Just worry about using it. Okay? So now you're a lot more happy with that. Okay. Any other questions with it? Okay. So we have cleared chapters 1, 2, and 3. 1 was kind of just, in my opinion, superfluous. 2 was looking at acids and bases. 3 was looking at our amino acids individually and how they applied back to chapter 2 with our acids and bases. Chapter 3 is now saying we've got amino acids, and what do we do with them?
Combine them.
Combine them to make
Poly.
polypeptides, and we take it a step further than that. We're really looking at now proteins. Okay? So we started with, hey, this is acid-base stuff that you theoretically should know, and arguably were never taught. That's fine. Moved into amino acids to say, okay, let's apply that knowledge to scale it up. How does that acid-base chemistry affect the structure and charge? And now what are we going to do? We're going to take that information and we scale it up one step more and say, what happens when we now put it all together? How do all these things then interplay with each other? The biggest part of how they interplay is going to come back to our intermolecular forces. Okay? So we need to remember the roles of our intermolecular forces. Okay, I don't remember. Did we see this slide yet? Okay. So we're vaguely familiar with our forces. So if I drew out, let's say alanine, why not? Middle of a protein chain, And since it's in the middle of the protein chain, do I care about the amine or the acid functional group? No. So all I'm going to look at would be the side chain, and the side chain was
Methyl.
methyl. I will probably keep looking at you right for that, just so you know. Okay, so we have the methyl group there. If this is now part of my protein, where would I expect to find this methyl in the protein?
Inside.
Why inside the structure?
Hydrophobic.
Okay. It's hydrophobic, okay, well, so what? What is 80% of life? Water. All of our cells are surrounded by water, which means if I look at that side chain, I need to evaluate its bonding structure. Carbons and hydrogens are covalent, which means London dispersion forces. I'm now concerned how that part of my side chain interacts with my solvent. And since it's biochemistry, that solvent is water, which has polar covalent bonds, which generates hydrogen bonding forces. I'm now comparing hydrogen bonding to London dispersion. Are they similar? No. Which means what do? They repel. Okay? In a standard organic system, what happens? We get 2 layers. Well, if we have multiple layers in our body, that becomes problematic. Okay? So we can't repel out into a secondary layer. So instead, we have to repel away from as best we can. Okay? And that as best we can usually means that that structure is going to get as far away from water as possible. The only location it has is to go inwards. So when we look inside a protein, we tend to find a lot of hydrophobic amino acids because they are scared of, that was a bit of excessive, repulsed by, also kind of extensive, but it's the more appropriate word, water. So we introduced the term of hydrophobic and hydrophilic. Okay? Why do we need a new term? Science. Okay? It's the exact same forces that we've been discussing. There is no difference. We're just now adding a new term to it because now it's easier to call them a new thing. Okay? So when we think about our amino acids, that's why I said when you memorize your 5, you want a smattering of structural effects. Because that smattering of structural effects then means when I ask where would you expect to find that amino acid, you can say that's on the inside, that's on the outside. What amino acid might we expect to find on the outside? Okay, glutamic acid, is that G? Sure. I think it's GLU at least. I got that 3-letter code right. And our glutamic acid, Mark, you going to back us up on that G?
You gotta have one more C.
Oh, there's another carbon in there? Is it 3 carbons or 2 carbons? In our structure? 2 carbons?
Aspartic is 2.
I thought aspartic was 1. How many carbons in that R group? It's 2 CH2s. So what you might be remembering, Ray, is the number of carbons, 1, 2, 3. Or you're right, and everybody's misreading it. That's fine too. Okay. Why would we expect to find that one on the outside of our amino acid? Or sorry, protein. Hydrogen bonding. Okay, take it a step further. If we're at physiological pH, did I draw the structure right? No, because the pKa of that is ballpark 2. Okay, and physiological pH is 7. So I'm more basic than my pKa, which means I need to be deprotonated. I remove the hydrogen, and the structure becomes negative, making it even more hydrophilic. I would expect to find glutamic acid on the outside, or glutamate on the outside of a protein, because it wants to interact preferentially with water. Or we could look at it in the opposite. It wants to preferentially avoid the hydrophobic interior. What's in the middle? Well, that was all our hydrophobic stuff. So we get multiple kind of levels behind this. So we have to be able to interpret and think about our intermolecular forces and how they contribute to our overall structure. Okay? So protein divisions, and this didn't work last time, so let's give it a shot. We've got our ATP synthase over here. What do all the different colors mean? Different proteins. Okay? So when we look at ATP synthase, it's not one protein. It's 1, 2, I think there's actually 4 or 5 in there. They didn't do a good job coloring it. Let's just say 3, 4, 5, 6, 7, 8. We've got 8 different proteins that have all self-assembled to make ATP synthase. If we look and break this down and look at just this outside part, that's what it would look like. Notice again the different colorings. Why? Different proteins. We look at the bottom part. Again, different colors because different proteins. If we break the bottom part down further, we notice that it actually breaks down into something like this, and we're now at a single protein. Okay? This is where the protein data bank can come into play. I'm actually not going to show this on the recording. For those of you watching the recording, you can click the link to our primary structure. The primary structure is just the sequence of amino acids. Okay, that's it. They'll be connected via the amide or peptide bond, which we've talked about. That's making up the backbone of our protein polymer. Okay? Because it's our backbone, that's where we address that we lost the acid functional group, and we lost the amine functional group, and we end up with the amide. The amide functional group does not have acid characteristics. It does not have basic characteristics. Okay? Which means I don't have to worry about pH changes affecting the protein backbone, which is probably a good thing, because if we change the pH, that could destroy the backbone of the protein. That's kind of a problem. Okay? What happens if we change the order, type, or location of any of the amino acids in that backbone? It becomes a different protein. Okay? Just like if we change, say, my hair color and sex, I may no longer be Mike. You might identify me as somebody else. Okay? Michelle, nice touch. Okay? So the same thing's happening within our proteins. Okay? Very subtle change, and this is the next part that's kind of fascinating, is a single change. If I remove one of those amino acids and replace it with a different one, okay, I could even go from something like alanine to threonine. Right? They're still both nonpolar proteins or nonpolar amino acids. But the threonine has a significantly larger side chain, or R group. That significantly larger R group is then going to prevent the proper packing of our protein, which then means the protein may no longer function the way it's supposed to. Okay? So the primary structure does lead to a lot of information. Okay? That's why we need the common classifications. Okay? This slide seems really oddly located. This should have been in the last section. I must have slid it down. This slide kind of goes through and summarizes the structure. You get your name, your 3-letter code, your 1-letter code. Notice in the purple, or the whatever that is, that pink, they're showing the backbone, and then we're showing the structure on the other side. So it's another version of the amino acid sequences that can allow you to see the functionality a little bit better. Okay, so depending on what you're trying to memorize, you may want different parts. I think this one works pretty well, because you get to see that, the R group, okay, and it's highlighted as separate because of the bluing. Close enough. Okay. So this is, again, where structure becomes important. If we go through and look at our primary structure, normal amino acid sequence, okay, for red blood cells, I forget which actual sequence this is. If we look at amino acid 6, and we have glutamate, and we make that amino acid valine. Okay, so that's all we did. So glutamate, there we go. There's our side chain down at the bottom. Unless we didn't circle that. So our glutamate's there versus valine, which is up here. That's all we did. It's the only thing that changed. What happened to the red blood cell? Wow. single amino acid, we go from nice, happy, normal, fluffy looking red blood cells to these wicked, jagged, stabby things. Okay? And the wicked, stabby things end up clumping up in your bloodstream and cause problems. Okay? Also known as sickle cell anemia. Okay? So that So that subtle change in our structure can make a huge impact on the overall thing. Okay? So we have to be aware that when we're looking at protein backbones, it's not just, oh, what does the overall structure look like? It can boil all the way down to a single amino acid. Okay? Which is why we need to talk about the single amino acids and scale our way up. Kind of make sense? 30 seconds. We kind of started to talk about this. Our protein backbone, notice that we're looking at 2 amino acids, not linked nicely. Okay. So if we try and highlight our amino acids here, we have really, there's 1 amino acid. Here's the kind of tail end of a second. That's part of that one. And here's the third kind of going out the other direction. Okay. When we go through and look at this, what they're trying to do is say what can happen. So the amide bond, okay, which is our purple right now, and the one I didn't highlight up there is green. Okay, that's between the nitrogen and the carbonyl. Nitrogen and carbonyl. We said we had limited bond rotation there because?
Resonance.
Resonance. What about the bonds on the inside? Did you say sp3? No? Okay. Black carbon's sp3, so I mean that was kind of useful. Those are what type of bonds in red, or on the inside of that? Those are sigma bonds. Those being sigma bonds means free rotation. That free should actually go into quotes because we have a structural effect on the other pieces that can sometimes prevent those rotations. Because we have the option for rotation, we can reference the angle at which it's rotated, believe it or not. That gets referred to as psi and theta. Okay, thank you. And those get named separately as the Ramachandran angles. Come on. Okay. We could technically quantify all of those. What we just need to realize is that we do have that rotation, and that does have an effect on our structure. We will pick up back on this slide when we look at our forces and our bond. Well, we did already talk about the bond. Okay, so we'll pick up there on